I'm Dick Moberg, and for more than 40 years I've been developing technology to advance our understanding of the injured brain. I've had a chance to work with some of the leading minds in the field of neuromonitoring, including physicians, researchers, and entrepreneurs. I want to share their stories with you in the form of a weekly podcast so you can stay current on the latest developments in the field and the innovative people behind them. This is my neural network. Hi, I'm Dick Moberg, and we're here at the AES meeting in Baltimore. And I'm here with two friends of mine from Vienna, Austria, two people I've known for quite a while. And they're here from the company Encevis, working on some really cool EEG analytics. And I'm here with Franz Fürbass, who's the chief scientist there. So welcome, Franz.
So, hi. Hello.
And I'm here with Anna Skupch. I mean, she has one of those names that's a little hard for somebody else to pronounce, but I know her as Anna, and I've known her for many years. She's a wonderful person, and Anna sort of is the glue at the company that makes everything stick together, and she does a lot of the marketing and all around fun person. And so welcome, Anna.
Thanks.
Cool. So I wanted to first start out about how did you get, tell us a little bit about Encevis. I know it's from the Austrian Institute of Technology, supported by them. And then how did you get into this area of EEG, analytics? So who wants to go first?
Okay, I'll go first. So we all started with a project that was in epilepsy. And we started with the seizure detection. And that was quite successful. We ran a prospective multi-center study, and the result of this seizure detection was so good that we made out a product. We added features as, for example, a spike detection, and then we moved on since we had this expertise in the EEG field, and since we always run our projects with clinics, and so we wanted to move into the ICU, and there is where we developed a new product for the ICU.
Yeah,
do you want to?
That's actually where we met Moberg.
Actually,
so when you go to the ICU, the Moberg was first coming to us. Hey, you have a nice product, and you do something with the ICU, and we want to hook up with you. That was a nice talk actually in the beginning. So I first thought that would be a nice idea to adapt the standard. So there was this new standard out from the ACNS. They just released it, and I remember it was the first year when the standard came out, we were renting around with some paper and showed it to Suzette LaRoche and say, hey, we have a user interface already for your new standard. And she was puzzling and looking at me, oh, you already have something for that. We just released the standard, and they kind of expected it to be accepted in 10 years, which is now, but it was kind of a little bit of early, I think too early for the market.
Yeah, I remember those days with Suzette. She was one of the pioneers with this, and Larry Hirsch.
Yeah, true.
Yeah, sure. That was really cool. Yeah. No, and I think you guys did an automatic detection or segmentation of the EEG into those classifications, right?
Yeah, that's true, yeah.
And I think you're the only one that does that, right? It's
until now, I think we are. So we are the only really FDA and CE release product that does the segmentation according to the ACNS standard. So we classify these periodic patterns, rhythmic patterns, this main term 2 basically. And we also did some main term 1 things like localization of these patterns. And we did a prospective study also in a neurological center of Rosenhügel and in the Medical University of Vienna, to test this and to find out the specificity and sensitivity of that. And we also then looked into how many electrodes we want to have in the ICU. Does the sensitivity really reduce when we reduce the electrodes? And that was interesting because it was the first study systematically looking into if you remove some electrodes, are the periodic patterns detected anymore or are they on? And we found out that the periodic patterns are quite sensitive to that. So you need a lot of electrodes, a lot of coverage in the ICU to get these patterns. And so this was the first study for that. And other things like burst suppression, they are fine with a few electrodes. So there's really major differences.
Yeah, and in the ICU, there are oftentimes electrode problems. I mean, obviously with patients and with putting in probes and keeping them on. So it's challenging from a number of aspects as well. So that's...
talking about artifacts, one of the main developments, very important development that we did was the artifact reduction. We call it PUEG. It really cleans. It's really something that I got so used to it that whenever I see EG somewhere and I have this instinct to click on the button where it says, in my viewer, it says PUEG, so to just take away all the artifacts. And so you just switch on this artifact reduction and you just have a really clean EG. So EMG, electrode artifacts, all kinds of artifacts. And that's always part of the pre-processing in R. So that's why our algorithms are so strong and the software is strong, because also we have a good artifact reduction.
And there's actually a funny story behind that, because in the project, the one colleague, he developed that. He initially was supposed to do it only for pre-processing. And in the end, it was coming out after some months, hey, we can show it really on the EG itself. So that the people can click on it and switch it on and off. And they said, oh, okay, anyone needs that? Is that really the need for the patient or for the clinicians? And in the end, it turned out it is a need. So they kind of always puzzle with these artifacts. Many of them, they say, okay, we are scared of this artifact, look through that or so. But when they have it and to switch it off, it's a different thing. So in the end, we left it in. So also for processing and for review.
Sure. And then again, that's also very challenging in the ICU artifact, unless the patient's in a coma. So those are the good patients.
Yeah. Well,
But I mean, there's so many other things that can happen in the ICU with the nurses, the manipulation, and all that. So it's it's a very challenging area. You guys have done, I think, some of the best work in that area. So moving on from the ACNS classification, let's talk about some of the other things. So certainly the seizure and spike detection is you've done a lot of work with that. And how's that going? Let's get a little update on that. And we'll talk about some of the AI work.
Oh, right. Yeah. In the last years, all this deep learning stuff really was amazing. Yeah. So people started with it, and in the beginning, I didn't believe that really have a benefit for us, because I always thought the amount of data we have is too low. So the first work that were coming out 2004, 2010, there were about 100,000s of data sets what they had in image analysis. And so I thought, okay, we have about 60 patients. What can I do with deep learning? There's no way to do it. And there was a conference, they asked me, did you do deep learning already? And I told them, this will never be done in the EG. We have 50 patients. What do you want to do with that? But now, in the last 4 years, we really managed to kind of train these networks with a few amount of data. So we use few amount of data that is labored, and the labored data is the problem. So labor data is really expensive. So people have to look through the data to mark something like a seizure, like a spike. And the second one has to do it. And they have to commit that this is really a seizure or a spike. And we found out we can also use unlabored data. So we use any data for training to improve the stability of the network. And now with our new spike detection things, it's really a gap. So we made a kind of a jump in the sensitivity. Mm-hmm. So like before, it was kind of weird thing to detect an epileptic spike. The field is really difficult. Every patient is difficult. But now with the trained data and with all these labors, we managed to reach 90% sensitivity, which is kind of unique on the market now.
Sure, it's with a small data set. I mean, any progress like that with small data is wonderful. I mean, that's great. So,
yeah.
So what else is new? What are you showing here at the AES meeting?
Well, we're very excited that we will have a new solution for the neonatology soon.
Great.
And so we have our best brains behind that. And the idea is to improve the usual monitor that you have with amplitude integrated EEG, with interburst interval, and also these measures to make them really accurate. The idea is to scan the EEG on one side to do an automatic classification of segmentation and classification into these patterns of tracé continu, tracé alternant, tracé discontinu, all these things that you have to go through page by page and check and find the patterns, and then kind of remember when they were and how often did they occur. And this you will have on one trace, and you will be able to show like 3 hours to 24 hours of results on just one screen for this classification. And then one thing that is really new is an estimation of the maturational age. So there, this group, the Cork and Helsinki group, that came up with the idea, since the age of the neonates can be recognized based on the EEG. Some people that have experience, they can recognize that. Instead of doing it visually, to do it automatically. So we have been using the neural networks technology and have with really good label data, we managed to train it on healthy patients in order to have on one side, when you record the EEG, and you immediately have an estimation of what, let's say, based on the EEG, what is the age supposed to be. And if the little baby is older, then you have to question that, how mature is the brain on one side, and then you can also follow the development. That would be the idea. So these are new measures that are going to be on the monitors that should make the review easier, and it should be also accessible by people that are not experts in EEG, and so also nurses and other people in the NICU.
And what I find most interesting in these techniques is sometimes you get a problem that you really cannot solve by handcrafted features, like looking into the signal and say, okay, what is the age? For me, it's really hard to tell. But with these deep learning things, they kind of extract this and compare it to the labels, and they find something that is really the age, and they do a regression and estimate the age. And there were other works that were amazing. For example, from Michel von Putten, he did the work on getting the sex out of the brain only by looking into the EEG. So you can tell if it is a man or a woman by only looking into the brainwaves. So you can also train a network like that. I don't know the clinical relevance of that, but in the end, it's interesting what you can do.
He's doing some amazing work over there in Netherlands. That's really cool. So good. So where do you see this going? I mean, I think, I'm a big fan of trying to automate this and seeing where it goes. I think the field of neurocritical care and EEG is just ripe for, getting a little more of the AI in it, and maybe a little less of the human. I don't know. There's so much variability. Well, in both of them now, but in human analysis, there's so much variability. But where do you see this going?
So I think one of the things we will see in the future is kind of biomarkers we see in kind of normal brainwave activity. So what's now the problem is you scan a lot of patients and you don't find the ictal activity, you don't find something suspiciously, and you're not sure what to do with the patient. It costs a lot to get all this patient in the long-term, hook it up in long-term monitoring. Perhaps with AI, we can find in small samples where you visually don't see any correlation, biomarkers that show you that this patient will have epilepsy in the future, or it develop epilepsy, or will get Alzheimer's or something like that. Yeah. So these are very recent approaches what we are looking into it, and I think this is really interesting for this deep learning stuff. I'm not really there to do that by handcrafted thing. I kind of rely on that methods to do that. And the other thing which is interesting, getting these things explainable will also be a challenge in the future. What you now get with all this deep learning is a kind of black box. So they tell you the age of the brain is like that, the detection thing is like that, but how you make it a white box so that people see where does this come from. So perhaps also for research to understand why is this a biomarker for epilepsy? Where should I look visually to see, okay, this one child would have a seizure in the next few days. So that would be interesting too.
Yeah, that's very cool. How about I don't know if they have this in Austria, but we have this thing called 23andMe, the genetic testing that where you can get your own genetic profile, and they have a huge, you pay like $90 and you'll spit in this little thing, you send it away, and they give you this, incredible genetic profile, and then they have one section is they can tell you the, if you're predisposed to certain diseases and all that, you can opt out of that, but you would think that combining that with some of your especially for things like Alzheimer's and some of these other things might be very, very interesting to do.
It is.
So, well, cool.
Right.
So, any final words on where you're going and what you're doing and
flying back tomorrow.
You're flying back tomorrow. Cool.
Coming back to the US.
Yeah, right.
Coming back next year to Seattle. I'm looking forward to the next meeting.
Yeah, sure. And I'm looking forward to my next trip to Vienna.
Absolutely.
I love seeing you guys there. It's a lot of fun.
We want to get you skiing. Right.
Snowboarding.
Snowboarding. Sorry.
No, and Austria is one of. I've only snowboarded in Innsbruck.
Okay.
But some of the other places I'd love to go. So I look forward to seeing you there.
Yeah.
Okay, you too.
Thanks so much for doing this podcast.
Thank you for inviting us.
Cool.
Thank you for the invitation.
Thank you. Good. All right. Thank you.
Bye-bye.
Bye. So thanks for listening to this week's podcast. If you enjoy these interviews, please take a moment to rate and review this show on your podcast app of choice. Subscribe to Dick Moberg's Neural Network to receive notifications when future installments are available. Thanks for tuning in, and we hope you'll join us again soon.